I first met John Singer some 39 years ago. He had actually been here for a while, and I arrived as a young fellow and a sort of senior postdoc. And as many of you know, this campus up here had not yet been constructed. And all these luminary professors like John had been recruited from around the land and were put down at the Scripps Institution of Oceanography, which was very nice for things like volleyball on the beach and other. But as far as the building resources were concerned, it was somewhat limited. Now, many of you probably don't know that John was in charge of the first building committee at Scripps, and it was constructed mainly at low tide. And the chemistry department actually had a set of pyramids nearby. But now, I show this only in a slight sense of levity here, but the introduction is rather a serious one, because beginning back in the early 1960s until now, John has in fact been something more than a pillar of the community. He was a great architect of the institution itself. Many of the people in the room today, I noticed, were in fact recruited in whole or part by John Singer. But at the same time, John was an extraordinary scientist, and we're in an era where most people don't really understand what their colleagues have done. We are all so narrow and niched that we hear things secondhand. Oh, somebody got promoted because they did something or other, whatever that is. But in fact, John has got a very, very distinguished record of scientific accomplishments. And I'm sure it's going to embarrass him, but I'm going to go over a few of them here this afternoon. Now, like others in the professorial circuit, John is well published. He has, in fact, published more than 300 high-quality articles. But the things that he did that advanced the field of biology are remarkable. The first item on this list is a paper by Singer and Schick, 1961. It was the introduction of the ferritin antibody method of labeling especially electron micrographs. You had in the 1950s tremendous successes with electron microscopy. People were looking at all sorts of cell bodies and the like, but now suddenly you could use this method and find out what proteins were in fact were existing on the cells and membranes of all these different things you were looking at. It was followed just a year or two later by the method of affinity labeling, whereby you could label antibodies, the active site of antibodies, or the active site of an enzyme, or anything that had an affinity for anything else. These are two fields that have lasted the entire 40 years in the interim. People are still doing this work. It's extraordinary that it started right with that one person. The next one is John Leonard, working with John in the 1960s. We're studying proteins as they existed in cell membranes. Now, everybody knows, because it's in all the textbooks, that John is associated with the fluid Mosaic model of the membrane. But it flows out so mellifluously, as though it was something you came in one morning and said, well, why might it be like this? But what people don't understand is that there were years of experiments and thought that went into that, including the use of the ferritin antibody method and other immunotechniques to decorate what was on the inside or outside of cells, and to how you had to understand the membrane in terms of this geography. Well, they continue right up down to the present. I've got many of the people in this room whose names are here. Dr. Takayatsu, for example, Mike Yaffe, others that were involved in always membranes and proteins, moving proteins across membranes, finding out how they got there, what they were doing once they got there. Now, I'm not the only one who has been impressed and took out what I thought were his 10 biggest contributions in the literature, because John has, in fact, earned many honors because of these. He's been elected to all the famous societies, the National Academy and the American Academy, and gotten lots of these name awards of Boris Pregels and Philip Levines and Hardens and Lonsteiners and others. He was an American Cancer Society research professor. That's a big deal. He was an all-university professor at the University of California. There are very, very few of those. People like Harold Urey. John was one from 1988 to 1994. Now, I think you know that in 1994, the University of California, again on its sort of cycle of hard times, went out luring people away, and John was among those who took what was called a VERIP. 1994 was the sort of the official end of being the university professor, but John continued. He was the faculty research lecturer in 1996, certainly one of the few that can be, at that age, be recognized by his faculty colleagues to get this very important annual award. One that's not on here is that in 1995, probably the greatest accolade that an academic can have is that more than 100 people descended on the campus from around the world, all of his former postdocs and graduate students, for a great feshrift. It was an occasion largely organized by people like Ann Dutton. It was just one of those things that you couldn't help but be envious of how many people had been placed around the world, Israel, professorships, heads of institutes in Paris, all over the country, that these progeny are, in fact, the reward for the kind of thing that John has been doing. Well, he didn't quit with the faculty research lecture in 1996. He, in fact, embarked on something that he's going to be telling us more about this afternoon, and that is that he wrote a book. The name of the book is The Splendid Feast of Reason, and it's a remarkable book. I think that if you haven't read this, you should go over to the bookstore and make them run out and order more, because it is John Singer. It's, of course, if it's John Singer, that means it's cynical. This is not a happy book. But there's plenty of wit in there. You'll recognize his wisecracks. The book reads just like John. But it's a serious thing, and it's something that he's really devoted the last part of his life to. We'll hear a little bit more about it, I bet, now. In the meantime now, I just hope that you'll join me in welcoming one of our most distinguished professors we've ever had. John, good to have you.
Perhaps the most important thing I can say in the first sentence. To me, the university is, or a university, I should say, is the prime focus of intellectual activity in the whole nation. And as such, its chief functions are discovery and enlightenment. Its main function is not to serve the business community. These days, it's getting a little hard to tell a faculty member from an entrepreneur. It's been that way for some time. That we have to live with, but those of us who are interested in the university as a place of the intellect have to keep the fires burning and making sure that that persists. Faculty members are mostly attracted to the university by the opportunity for discovery, and in particular, the chance to do research. Some of them are also very much interested in teaching, but in our kind of society, in the university, research and teaching are equally responsible activities for the faculty to be involved in, and I'll have something to say about both. I want to talk first about research, since that is always our chief interest, and something about styles of research, which I've come to understand over the years since I've been doing it, can be quite different for different people. One style I've sort of diagrammatically sketched here is what you can call the straight arrow approach. You start with a field that at some early stage is a very little diversification. There's very little known about it. You plunge in, and as time goes on, lots of more things are learned. The system itself that you're working on gets a lot more complicated. And you yourself wind up, after 10, 20 years, taking a larger fraction of work in the field, but it's still a small fraction of the total activity going on at that time. That's a very difficult thing to do over a period of 40 years or so. And some people have been extraordinarily successful at it. I think of Arthur Kornberg, for example, studies of DNA chemistry, as somebody who has always been at the top of the field and has managed, despite its tremendous diversification, to be a giant in the field. Most of us do this kind of straight arrow work, and I'm afraid it's going to be more common as time goes on. It gets harder and harder to break out of the field in which one has expertise. I realized sometime 30, 40 years ago that I wasn't inclined to do things this way. I was going to be more, should we say, a jumper from one field to another as I saw fit to make the change. I started out life as a protein physical chemist, and I knew what was known all about. I knew all about what was known about proteins in the 1950s, when the whole idea of hydrophobic interactions and the way in which soluble proteins were put together with their hydrophobic residues inside to satisfy the hydrophobic interactions, and the charge groups on the outside of the molecule to interact with the water. And I could have stayed with that. Many people have stayed with that area. It's still a very vibrant area involving a lot of X-ray crystallography and studies like that. But it's still, of course, not clear what the predictability of protein structure is. So that's still a viable field. But I got out of it fairly early and moved into studying antigen-antibody reactions, which I picked up a little bit of while I was a postdoc with Linus Pauling. And the problem in that field was that the mystery, which is sort of at the bottom of all of this to begin with, the mystery was absolutely unclear. How one could have antibody molecules of millions of different specificities when you had only a limited number of genes apparently devoted to the production of these proteins. And we worked on it for many years. Russ mentioned we did the affinity labeling studies. Leon Wofsy and I invented the method to try to study the active sites of antibodies, in particular, to try to see what they were like and so on. But in 1965, a revolution really occurred in the field when it was demonstrated that antibody molecules had a constant region and a variable region, as you all well know. And this opened up the whole mystery as being due to, ultimately, to jumping genes, that the antibody molecule was put together by genes coming together to fashion a tremendous multitude of protein molecules that couldn't otherwise be made. And there was, in 1965, then just the beginnings of all of this work that took place over the next 15, 20 years. And one could have stayed in that field profitably for a long time. But at that particular juncture, I felt the mystery was gone, and it was time to move on to something else. And that something else was something that had been sort of festering in my mind for a long time, and that was membrane chemistry. It's very, very difficult for you, it's difficult for me, to realize what membrane chemistry was like in the early 60s. No membrane protein, no integral membrane protein had ever been isolated, identified, or characterized at that time. And we had really, literally, this picture of what a membrane was. It was this famous Jeffson Danieli Robertson model, where the lipid is organized as a bilayer, but the proteins were completely, they didn't know what to do with the proteins, so they picketed the lipids on either side with them to get the kind of thicknesses that were seen with membranes in the electron microscope. But next to nothing was understood. Now, as protein chemists, we were very much aware, as I mentioned, of these matters of hydrophobic interactions, and every protein chemist was. But as it turned out, not every protein chemist got interested in membranes, but we did. And the result was, with the application of what were standard ideas in protein physical chemistry, we eventually came up with the model that you know about, the lipid protein Mosaic model, which I'll have something more to say about. In this model, what was new about it was that we proposed that membrane proteins traversed, many of them, traversed the thickness of the membrane, protruding from both sides, but that the interior portions, which were associated with the hydrophobic lipid interior, would be predominantly hydrophobic themselves. That is, all the amino acid residues in that domain would be hydrophobic. The exterior regions of these proteins would be much like ordinary water-soluble proteins in the way in which their amino acid residues were distributed. Well, this is all, of course, old hat, but at the time it was a rather new way of looking at things. And we had a few years to spend at our leisure sort of inventing what membranes might be like and what the membrane proteins might be doing and how they might be organized, because it wasn't still for another several years that the first integral protein of membrane was isolated. This next slide illustrates some of the thoughts we had about membrane protein. First of all, we thought there were probably two different kinds. The integral ones that you saw in the last picture, traversing the membrane, but also proteins that were loosely attached to the membrane, which were in danger of being regarded as typical because they were easy to get at the time. Cytochrome C was an example. You could knock that off mitochondria with 3 molar KCl, And everybody was studying that as if it was a typical membrane protein. But from our perspective, we recognized that this represented a separate class of proteins that were attached probably to the regions on the, that protruded from the membrane. The peripheral proteins would be attached here or here, and more or less typical weak protein-protein interactions would be involved in them, but that the peripheral proteins would look just like ordinary water-soluble proteins otherwise. So there was this distinction we drew between the two kinds of proteins. We made a big point of the large cost and free energy that was required to bury charged groups or residues that were capable of being ionic. And that for this reason, we expected them to be excluded from the membrane interior. Also, because these interior regions of the membrane would be not exposed to water, the peptide hydrogen bonds would have to be made in order for the free energy of the resultant structure to be satisfactory. But the most precise way, the most common way that they could do it and most efficiently was as alpha helices. So we predicted that the transmembrane domains would be largely alpha helical. Again, remembering there was none of these proteins around to compare with. Furthermore, and I'll have more to say about this a little later, the notion of transport through membranes of ionic and small hydrophilic molecules was in a state of disarray. At that point, the general idea in the early 60s was that there would be proteins in the membrane called carrier proteins, which would pick up an ion like sodium or something, rotate around to the other side of the membrane, and deliver it to the other side. Now, we recognized that this was thermodynamically out of the question. So we decided that the more likely possibility, which I'll talk about in a little while, is that you had specific types of proteins that would be involved in transport activities, which had channels running through them all the way from one side to the other, channels that were filled with water and that could therefore be lined in the channels with ionic residues as required. Now, with regard to this question of ionic groups inside hydrophobic proteins, inside hydrophobic milieu, there was, and unfortunately there still is, a lot of, shall we call it baloney, that suggests that it's possible to bury ion pairs, plus charge with a minus charge, inside hydrophobic domains, that they would be stable in those situations, and therefore that they wouldn't be excluded, ionic groups wouldn't necessarily be excluded from these interior regions of proteins. Well, we just looked up some old data on the solubility of glycine in different organic media, glycine being one ammonium ion and one carboxylate ion, an ion pair in the molecule glycine separated by a methylene bridge. And as you can see, the solubility of glycine decreases sharply with the increasing non-polarity, with increasing hydrophobicity of the solvent. Even when the solvent is polar as acetone, it takes 6 kilocalories per mole to transfer an ion pair from water to acetone. So to a hydrocarbon, which has an even lower dielectric constant, this number I estimate it to be more like 15 or 20 or whatever. And it's big enough so that you don't bury ion pairs in hydrophobic media. We also sort of guessed at what might be the arrangements of these unknown proteins in membranes, and I suggested 3 types. One where you had a single transmembrane pass, which was very hydrophobic, all hydrophobic residues, but had these structures on both sides. One where there were multiple passes of largely helical domains extending from one side of the membrane to the other, tied together by loops. And these were the proteins I thought about as possible transport carriers, that they were composed of subunit aggregates, maybe of subunits that were quite homologous to one another, but that in forming an aggregate, they also formed a water channel. And through this water channel, the hydrophilic ions could travel. So this was from another more artistic person who represented what we were thinking of. This would be a tetramer running all the way through the membrane. And the idea was that in the interior of the membrane, an ion binding site might be conformationally rearranged by some appropriate free energy change accompanying the binding of some substituent, and that in this manner a small ion could be transmitted from one side to the other. Now, I hate to take all your time on this because this is obviously old hat to everybody in the room, but I do take a certain element of pride in it, in the sense that there was no data at the time to warrant these suggestions, and it's only more recently that these suggestions have come to be shown correct. Another question had to do with the symmetry of membranes. If our picture was correct, it wasn't really feasible thermodynamically for molecules that were on one side of the membrane with their ionic and polar groups to rotate through the membrane to the other side and achieve an equilibrium distribution across the membrane. And we tested this idea with a simple experiment through an erythrocyte ghost, which we could peel back and reveal the inner surface of the membrane and the outer surface on the same specimen. And now we could use some labeling reagent. This is work that Garth Nicholson did. He attached ferret molecules to lectins, which are proteins that bind to sugars. And he got these results that these are all the ferritin lectins here on the outer surface, nothing on the inside. So that the sugars on cell membranes, which are predominantly on the protein moieties, but also on some of the lipids, are all on one side of the membrane and not the other. Now, at this particular stage, of course, if proteins are asymmetric in membranes, some asymmetrically located, we showed this was true for other kinds of proteins. This has much to say about how membrane proteins get inserted into membranes in the first place. They don't get inserted randomly from both sides. They must get inserted from only one side and then stay that way in their orientation in the membrane. So we didn't do anything much about it, but we anticipated that there was going to be some kind of directional insertion of membrane proteins in the process of getting proteins into membranes. Now, finally, about 7 months after our papers were published, outlining most of this kind of detail, Marchesi and his co-workers characterized the first integral membrane protein in the tail. This was glycophorin A from the red blood cell membrane. And it was a molecule of about 130 amino acids that had the characteristics that it had from the amino terminus on sort of regular sequence of amino acids. And then at this point, a stretch of about 20 amino acids that were all hydrophobic. And then another stretch that were a mixed type. And it was very clear that this was the hydrophobic stretch that we had predicted would be present in such integral membrane proteins. And the saccharide residues on this protein, which is a glycoprotein, are all on the amino terminal side. There are none on this carboxyl terminal side. So this part is sticking out of the membrane into the exterior, and this part on the interior. There haven't been many proteins that have been studied integral proteins have been studied extensively because they're very difficult to crystallize in appropriate form for X-ray crystallography. But 15 years later, Diesenhofer and his co-workers managed to crystallize the photoreaction center of bacteria, which consists of 3 different integral proteins in a complex. One of these, in the middle, is a single transmembrane protein. The other 2 are homologous 5 transmembrane proteins. And there's in addition a B-cytochrome peripheral protein associated with this complex that I'm not showing here. Anyway, when the 2.8 angstrom resolution X-ray structure was shown, this was the model. The alpha helix, it's all alpha helix inside. The helices are very nice straight bundles. And if you look for the charged amino acids in this structure, these are the arginines that you can't see. They're just all over the place, labeled on the region up here and up here. And essentially, the interior region is empty of the charged residue arginine, and this is a lysine. The same thing. The charged residues are not there. So all the predictions that we've made about the hydrophobic interactions in the membrane were pretty well substantiated by this model. Not all membrane proteins are going to be as simple as this, in which the helices are nice and straight and parallel. Recently, a few years ago, rhodopsin crystal has been investigated. This is a stereo picture, so pay attention to only one side of it. The main point of it is that the helices are angular with respect to the plane of the membrane. They're kinked, and they cross each other, and it's more than likely that this type of more complex structure is going to be more common among a lot of different proteins. But we see what happens is that you now create crevices in the molecule by virtue of how the helices are placed, and these can allow water molecules to enter into what would be normally thought of as the interior portion of the membrane protein. And now you could have charged groups in here and a lot of other complications that ensue from having these more convoluted structures of membrane proteins. Finally, about membranes, what I wanted to say is that from the beginning, when we had this lipid mosaic model in mind of how the proteins and lipids were organized, we asked ourselves the question, could they move around? And what was to keep them from being mobile in the membrane? The lipids were known to be fluid at body temperatures, and we were at a seminar at Rockefeller, I remember, and I asked this question. Somebody came to tell me that there had just been a paper published in the Journal of Cell Science by Fry and editing. Now, I had gotten as far as being a membrane chemist, but I wasn't a cell biologist. And I never read the Journal of Cell Science until that particular time. And in their experiments, schematically, they did a very neat experiment. They made a cell fusion hybrid of a human and a mouse cell with different antigens on their respective surfaces. And what they found was that immediately after fusion, by fluorescent labeling the respective antigens, they found that they remained localized to the two sides corresponding to the fused cell, but that within 40 minutes, all the antigens were mixed, indicating with controls that they showed that there was global diffusion, not just local diffusion, but global diffusion of all of these molecules in the cell membrane, so that the model became the fluid mosaic model, incorporating this finding into what was the natural structure in which this diffusion could be accounted for. At this point, there was still obviously everything left to do in membranes. Very few proteins by 1970, 75 had been characterized, and the age of DNA chemistry was just beginning, recombinant DNA, and there was plenty to do. For good reasons or not, I don't really profess to tell you, I decided to make another jump at this stage before we really had that much more information. The reason was I'd always had this secret ambition to be a biologist. I was from the time I was in Pauling's lab. And he wasn't a biologist either. He was, I didn't know that. He was a physical chemist dabbling in biology. And really, that's what I had been for all this time, through the whole membrane story. It seemed to me that this was an opportune time to get into cell biology. I had the tools. I knew about what the outside of the cell looked like. Now I wanted to know what the inside was like. And we had another tool for that purpose, which Russ has already mentioned, and that was that I had developed in 1958 a ferritin antibody method to bring fluorescence microscopy that Al Kunz had devised to the level of the electron microscope. And here's a picture that I took myself with an electron microscope that I was using at the University of California at Berkeley on my sabbatical, of tobacco mosaic virus particles coated with the ferritin antibody. It was a ferritin of these little black dots. I felt at the time that this was going to be a very valuable tool in cell biology. But what was needed was to be able to stain ultra-thin sections of cells with the ferritin antibody, or as subsequently became more popular, gold conjugated antibody, because gold has a higher electron density than ferritin. Now, all the commonly used ultra-thin sections that have been in microscopic use for many years were plastics of one kind or another. And ferritin antibody stuck to them, all of them, indiscriminately. At that point, fortunately, Dr. Kyotaro Takayasu joined our laboratory and set on the problem of making ultra-thin frozen sections that could be used for doing this kind of work. Now, if you freeze water and try to cut a 100 angstrom section, it cracks, it's brittle. So he had to work out a technique for making ice cut in 100 angstrom thick dimensions, which he did in 1973, and that's been a centerpiece of many of our studies on the interior structure of cells using this methodology. We did a lot of work with it, but I'm not going to have the time to spend a lot on it. We did one of a number of experiments that will interest certain members of the audience. We were interested in the enzyme HMG-CoA reductase, which is the key enzyme in cholesterol metabolism. It's the target for all the drugs, the statins, that are used to control atherosclerosis and so on. But at the time we undertook this work, nobody knew where this protein was. It was supposed to be all over the cell. We showed by Tox technique, and this is the work of Gilbert Keller in the lab, that it was to be found in peroxisomes. This is gold labeling. Peroxisomes labeled for this HMG-CoA reductase, but mitochondria, RER, any other of the organelles were unlabeled. So in this manner, it was demonstrated that the principal site of cholesterol biosynthesis, which is, of course, a vital part of cell physiology, was taking place in the peroxisome, which was then a very obscure organelle in the cell, made prominent later by Suresh Subramani and his co-workers. So this was work that was of some considerable interest. We did a lot of work on muscle structure, on the cytoskeleton, on cell motility, a whole host of other things that occupied us for 20 years. It never exhausted us. We didn't have any reason to look for something new to do, because it was always there in the field of cell biology. Though I should add that in recent years, I've been fortunate enough to have a colleague from the medical school, Dr. Nazneen Dooji, and visiting in the lab, who taught me something about Alzheimer's disease, which I didn't know anything about before, and which we have been collaborating on for a number of years with useful results. And this might be constituted another jump in my particular research career, but it's certainly going to be the last one, I'm sure. I don't know which way to recommend to new people in coming into biology, the straight arrow approach or the jumping approach. They both have their virtues. I think it comes down to the personality of the individual research worker. It's getting harder to jump one field to another because there aren't many fields left that still have big mysteries in them. And the tendency is to stay in one area where you can be happily funded. The NIH takes great pride in funding only experts in a field, so the chances of getting funded on something you're not well published in is next to zilch. So the way we did it was to bootleg it on other grants. And that still is the best way to do it. Another way is to take advantage of sabbatical leaves and go to some place where somebody's doing something quite different that you're interested in and find your way into that kind of field in that manner. But there are these two ways of doing things, at least, and what they bode for the future, I'm at some loss to say. I want to turn to teaching. Briefly, I've taught maybe 8 different biology courses in the course of these 40 years at UCSD, starting from freshman biology. I had the major biochemistry course for undergraduates for about 8 years. I had Marty Yanofsky suffer through one of those, as I recall. And fortunately, he managed to work himself into an A for the subject, which would have been catastrophic otherwise, because his father would have killed either me or him if he hadn't. I taught immunology, I taught cell biology, I taught membrane biology, and I enjoyed each one. Each one was for a period from 5 to 10 years time. By the way, it is the best way to do the jumping research routine, to teach a lot of different courses, because teaching has always been, for me, the sharpest way to crystallize what I want to know. If I have to teach something to a student conscientiously, I have to know what I'm talking about. I hope all of you feel the same. That takes a kind of learning that surpasses anything else, like just reading articles or anything like that can do for you. So, not the least attribute of teaching is its very good effect on one's research program. Now, I can't let you go without going on to another subject which involves teaching, but is in a way a very different kind of animal altogether. I've come to feel over these 50 years, starting at least 15 years ago, that university teaching is in great trouble. It's really quite ineffectual. It consists in each, and I don't mean just biology by any means, in every part of the university. The major or any other course of teaching consists of a whole series of discrete courses, one on top of the other, maybe 30 courses in the course of a major, I don't know how many. They're absolutely disconnected. Nobody connects them in any significant way. They're not what the student really needs. A student needs an integration of all of this tremendous amount of knowledge, not this fragmentation we give them. Now, why do we give them this fragmentation? It's in our image. It's our research image that's being projected onto the teaching activities that we perform. That's all it is. We teach cell biology, we do cell biology research, we teach cell biology, and vice versa, and so on. So, and this occurs in any department you want to name. I decided to do something small at the beginning about this about 15 years ago. I instituted a senior honors course in Revelle College. These were for all kinds of majors, but they had to be seniors, and they had to have at least a 3.5 grade point average so that they could cope with it. There were 16 students in each class. We taught 10 3-hour lectures each term, and they weren't really lectures. I was joined in this process after a few years by Professor Stroll of the Department of Philosophy, and between the two of us, we had a very remarkable tension going between the scientist and the philosopher, and we're very good old friends, so it never came to blows. And what we did was to pick a series of topics after introducing them, such as you see down here, that have to do with science and technology in civilization. That was the name of the course. And we talked about a lot of different things that you normally don't process in any course that I know about, and certainly not via a scientist. You might get some social scientists taking a crack at some of this, but they largely don't know what they're talking about when it comes to science, which they enjoy telling you they don't know anything about. So The students come in, 16 are selected for 16 different topics. They pick their topics. They are assigned to give a seminar for 45 minutes. For most of them, as it turned out to our great surprise, it was the first seminar any student, any senior had ever given before of any intellectual content. They give a seminar for 45 minutes, and then there's 45 minutes of free and open discussion. The attempt is to do something very different. And Avram and I are both approaching senility. The chances are very good that we will stop giving this course pretty soon. We hope somebody else undertakes something like it. We hope that this kind of a course not takes the place of what we're doing. We can't really do that. But adds at the end, not only in science and technology, but in the social sciences and the humanities, in the end tries to tie some things together for the benefit of the student who's about to leave, as I am too. Thank you.